పార్లర్ పోదాం అనుకుంటాం. ఎప్పుడు ఎక్కడ రోజు పెట్టాలి వాళ్ళిద్దరు పోతే అది గంట అయ్యే పని కాదు పార్లర్ పోతే మళ్ళీ వదిలేసి పోయినారు. తెలిసే మా నాన్న దగ్గర మాట్లాడింది సార్.
Satyasha madam, good morning Andy.
Sir, namaste, sir. Good morning, sir.
How are you madam?
Sir, fine, sir. Thank you. How are you, sir?
Fine madam. Thank you for the accepting of the invitation for our
My
FDP.
pleasure, sir. Thank you.
My best wishes to Principal Nagbhushan garu.
Oh, thank you, thank you. And also congratulations for you too, sir.
Okay. For what madam?
Sir, you have got NAC accreditation recently, just before we came.
Okay. Just exactly.
Yes, sir.
Yes, exactly. Thank you so much madam.
Thank you, sir. Thank you. Sir,
Hmm.
is my voice and my presentation clear?
Your voice is
Right.
audible madam.
Okay, okay, sir.
Shall we start madam?
Sure, sir.
Hmm. Hmm. Manasa madam,
Yes, sir.
you can start.
Uh, Good morning to the esteemed speaker, participants, Principal Dr. A. Lakshman Rao, sir. Myself, Manasa, Assistant Professor, Department of Pharmacology. On behalf of VV Institute of Pharmaceutical Sciences, excited to welcome you all to Day 4 of 1 week online faculty development program on Cell Therapy, Pharmacological Insights and Future Directions, organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru. I welcome Patron Dr. A. Lakshman Rao Garu, Professor and Principal of VV Institute of Pharmaceutical Sciences. Now, I would request Dr. A. Lakshman Rao sir to speak a few words about today's session.
Thank you, Manasa garu. Respected resource person of today's session, senior academician, Dr. A. Pratyusha garu, Professor and Vice Principal, Hindu College of Pharmacy, Department of Pharmacology, Amaravati Road, Guntur, Andhra Pradesh, participants from various pharmacy colleges. A very pleasant morning to one and all for the 1 week online faculty development program on cell therapy, pharmacological insights, and future directions organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru from 9th to 14th December 2024. In day 4, Dr. A. Pratyusha will deliver a lecture on reverse pharmacology. Reverse pharmacology is one of the novel topic in this particular current scenario. Just I'll give you the brief outline about the today's topic. Reverse pharmacology is an approach that involves starting with traditional medicines, natural products, or therapeutic practices, and then using scientific methods to identify and understand the underlying molecular mechanisms and bioactive compounds responsible for their effects. Unlike traditional pharmacology, which begins with the molecular target and developing drugs, reverse pharmacology works backward by validating the therapeutic potential of existing substances or practices. This approach helps in discovering novel drug candidates, elucidating mechanism of action, and improving the efficacy and safety of the treatments, often inspired by indigenous knowledge and traditional healing practices. And that is the brief outline about the today's topic. And on behalf of the management, principal, staff, and students, let me thank the today's resource person, Dr. Pratyusha Garu, for sparing her valuable time for our online FDP program. And thank you, one and all. Now I hand over the session to Ms. Gude Manasa, convener for the Faculty Development Program, for introducing the today's speaker to the participants. Over to Manasa Garu.
Thank you, sir. We are very grateful for your insightful words, sir. It gives me an immense pleasure to introduce today's speaker, Dr. Anna Pragada Pratyusha Ma'am is a dedicated academic and researcher, currently serving as the Vice Principal and Professor in the Department of Pharmacology at a prominent institution. She earned her PhD in Pharmaceutical Sciences for Acharya Nagarjuna University in 2021. Following her MPharm and BPharm degrees from Andhra University and Hindu College of Pharmacy, respectively. With a strong academic foundation, Dr. Pratyusha ma'am has been actively involved in both teaching and research since 2015. Her research interests include pharmacological screening, anti-inflammatory, antioxidant, anti-diabetic, and anti-anemic activities with more than 12 research papers published in reputable journals. She has also been graduated an Indian patent for designing a nano biosensor to detect glycated hemoglobin levels. Throughout her career, Dr. Pratyusha ma'am has guided over 12 undergraduate and postgraduate research projects. In addition to her research achievements, Dr. Pratyusha ma'am has been recognized with several accolades, including the Young Scientist Award 2024. She is also a qualified candidate in various prestigious exams such as ANU RSET, GPAT, NIPER, and Manipal University entrance. Dr. Pratyusha's ma'am technical skills include expertise in animal handling, pharmacological screening, and proficiency in statistical software like GraphPad, Prism, She is a member of several professional organizations, including the Indian Pharmaceutical Association and the Indian Pharmacological Society. With a passion for both research and mentorship, Dr. Pratyusha ma'am continues to make significant contributions to the field of pharmacology. Now, I would like to hand over the session to the today's speaker, Dr. Pratyusha ma'am. Ma'am, can you please take the
Thank
session,
you.
ma'am?
Thank you for your nice words of introduction about me. Uh, I would request all of you to bear with 2 minutes wherein I can get connected with other source of internet. I got interrupted from the internet.
Okay, ma'am, no
Just a
problem.
couple of minutes. I think I'm still online.
Yes, ma'am.
Yes, yes, yes. Right. Thank you. Thank you for your nice words and with a warm good morning and good wishes to all the participants here. So let's now, can I share my screen, madam?
Yes, ma'am.
Yes. So now let's look into the major insights of reverse pharmacology and its applications. Reverse pharmacology, the word itself, the title itself speaks about pharmacology in a reverse approach, where we start a pharmacology with a preclinical study wherein the activity already estimated or the activity already thought it would have either the new molecular entity or a certain extract of a plant, whatever the drug or whatever the extract it is, we have already an insight of what kind of activity it would have supposed to have. So with that approach, we go for preclinical trials. And then once we get established with a good evidence of safety and least toxicity, and after further processing of the dose calculations and dose designing, we then go for clinical trials wherein we use phase 1, phase 2, phase 3, and phase 4 of clinical trials. And then we launch the product into the market, right, in phase 3. Okay, post market surveillance in phase 4. So the concept here is the time taken for the entire process of this drug development is around 10 to 15 years, which accounts for about 1 to 1.5 billion per development of a single formulation. So here what the reverse pharmacology thrust is, it has a reverse approach for taking the evidence of the traditional medicine, which has already enough data procured so that it establishes the safety margin for its applicability for various margins of applications. So let's see the introduction of reverse pharmacology here. Reverse pharmacology is an innovative approach that shifts the focus from drug discovery to understanding the natural compounds and their therapeutic effects. During the development of a new drug, drug targets are identified. Many molecules are synthesized through various chemical and experimental methods, and the drug candidates are tested through high throughput screening and or binding to the specific molecular macromolecules. So here we go in a process from step 1, 2, 3 to 4, wherein ultimately the drug candidates reaches as a formulation to the community. Despite the advances in genomics, proteomics, and metabolomics, the drug development of a new drug is still costly and still time consuming and less productive. And the return on investment is of very low. That indicates like in spite of we spending lot and lots of years on one particular molecule, and still we invest much and much amounts on the development of a formulation, but still that turns out to be failure. So we, in recent times, we have seen many molecules that were first launched and were again withdrawn from the market for various toxicities they have elicited. So this has laid major foundation for the applicability of reverse pharmacology in the drug development scenario and process. So what is reverse pharmacology? Reverse pharmacology is a science of integrating bedside documented experiential hits into leads by transdisciplinary exploratory studies and to further develop these leads into drug candidates by the state of the art experimental and clinical research. So here we have to understand that it is an integrated science in transdisciplinary science where we have the major concern or major backup of all the documented evidence. Documented evidence in the sense all the all reverse pharmacology has a kind of major support system from its Ayurveda, or we can call as traditional medicine. Ayurveda in India and various other Asian and Eastern countries have traditional medicines where they have significant utility even now. Though we are at an era of using all synthetic medicines or allopathic medicines, we are still reliable. We have still reliable sources in our traditional medicines that are still being so beneficial at par to the allopathic medications. So that is the basic baseline for the development of reverse pharmacology here. Since it has a transdisciplinary exploratory studies for its baseline, we can consider the reverse pharmacology has safety as its prime motto. Since all the traditional medicines have already established for its safety, we can now use the process of reverse pharmacology for developing the applicability of the same candidate into a different or novel approach which can target other ailments, right? So the scope of reverse pharmacology is to understand the mechanism of action at multiple levels of biological organization and to optimize safety, efficacy, and acceptability of the leads from natural products based on relevant science. Coming to the historical context of reverse pharmacology, the basic foundation for reverse pharmacology was laid by Ramnath Chopra sir and Gnanath Sen sir. They were the lead members or pioneers we can call for the foundation of reverse pharmacology. As we all know, Ramnath Chopra Garu is the father of Indian pharmacology. So Ramnath Chopra sir and Ramnath Chopra Garu and Gnanath Sen Garu together worked with Karthik Bose to demonstrate antihypertensive activity of Rauwolfia serpentiana. Of course, now Rauwolfia alkaloids which were isolated from Rauwolfia serpentiana were not used for antihypertensive activity, but first developed as an antihypertensive agent. So that is how the study or the profile established from these sources helped us to redirect and reorient ourselves for the other applicabilities or novel applicabilities of the same existing drug. Dr. Ashok Vaidya sir is considered the father of reverse pharmacology for his extensive research that led to the paradigm shift in drug discovery. In recent times, we have to understand our own indigenous products from CSIR and NBA. CDRA Lucknow have launched many products like Picroside 1, Picroside 2 have shown a very prominent applicability for NAFLD, like non-alcoholic fatty liver disease, which has shown much significant effect protecting the hepatocytes when compared to the standard drug like Silymarin. We usually take Silymarin as a standard drug for all the hepatoprotective activity we study, right? So in that cases, there might Picroside 1 and Picroside 2 molecules which were studied from a complex Ayurvedic formulation. They were isolated and they was significantly established for its activity of proving much significant levels of hepatoprotection against the standard drug Silymarin. So all the study has been led by Dr. Ashok Vaidya, who has been coined with the term father of reverse pharmacology in India. So why do we need reverse pharmacology? So why should we think of reverse pharmacology and why do we need it? So basically, we have come to know that it is already established drug are now considered for its new applicability. So why should we think of it? Why not we go for the conventional drug discovery process using classical pharmacology? Because of the limitations or constraints by time, cost, toxicity, and the decreasing approvals for new chemical entities. So, if we see the general scenario or a conventional scenario of drug development, we'll first go for target identification, then lead identification, lead optimization, preclinical studies, then go for clinical trials. All this process is expensive, time consuming, and includes numerous bottlenecks. Of course, for a product to get launched as a successful formulation, it takes minimum of 10 to 15 years time. And you can't just rely on this entire process if it fails to establish 100% or more safety and efficacy in post-market surveillance. So these kind of bottlenecks are now addressed through the phenomenon or to the process of reverse pharmacology. So what exactly happens with reverse pharmacology? So in reverse pharmacology, we start with a drug molecule which has already enough data procured about its safety, efficacy, and its dosage regimens. This is all out of experiential data pooling. This is being done by all the practitioners, clinical practitioners like doctors. All these doctors, when they prescribe a particular drug to a patient, they'll have enough knowledge or enough documented evidence of what kind of effects it has elicited and for what kind of ailment it was prescribed and the duration of course given to the patient and all the profile they have already documented. So with that kind of documented evidence, we'll further explore, right? We'll explore the applicability of that particular formulation for a newer avenue, like newer activity rather, right? If that particular molecule is established safe, you can now use the same for other purpose of treatment. So that is the major intention of introducing reverse pharmacology. Since the data is much available about the drug, you cannot invest your time to procure the data again and again, which reduces the time and makes the drug available in the market within 4 to 5 years of time. That is the major advantage we are supposed to understand about reverse pharmacology. So the basic concept of reverse pharmacology has 3 domains, which includes lab to clinic approach. Okay, so lab to clinic approach is conventional method wherein a molecule is defined, identified, developed or extracted, whatever, either through the natural source or a synthetic source. A molecule is identified as a lead and this lead molecule is now experimented on animals using preclinical studies. Then it establishes safety and efficacy at preclinical level. Then it goes for clinical trials in a human. So this is conventional or classical pharmacology. So what is the concept of reverse pharmacology here is to clinic to lab, right? Clinic in the sense it was first prescribed to man after establishment of safety, and this clinical data which was given from the doctor or a practitioner is then identified for newer avenues in the animals, and once if it establishes a newer approach or newer applicability, then this molecule is subjected for further studies at lab for the development of similar molecules like their analogs for this and for the production of analogs with the same expected outcome, right? So reverse pharmacology has safety as its prerequisite. Coming to the phases of reverse pharmacology, it has 3 main domains like experiential domain, exploratory domain, and experimental domain. Like first is experiential domain. It covers the literature research, Ayurvedic pharmacoepidemiology, and well-defined, modest observational therapeutic studies with objective targets of response. So once if you have to identify a formulation or an extract or a drug molecule to be pursued under reverse pharmacology, you should have all this experiential domain collection at your hands. Like the complete research literature of that particular extract or molecule or whatever we have chose for our reverse pharmacological study, right? Majority of reverse pharmacology now till date is based vastly on Ayurvedic pharmacological products. So why not we pursue the same in allopathic medication is the trending approach now for all the academic researchers, right? We shall discuss that later. And now, in order to pursue the reverse pharmacology, we should first at hand have the complete literature of that particular extract or drug or formulation what we are trying to pursue further. So it should convey a well-defined, modest observational therapeutics. Like it should include all the documentation should be robust and it should have all the data which proves that to be much safer. So in order to develop this particular formulation into a newer application, we should first ensure it is safe in its original application, right? Once if it is safer for its basic and intended use, then you can modify it for further applicability in various new avenues or various new areas like other what we call as therapeutic applications. So then we have to explore the animal studies that cover indication relevant in vivo and in vitro studies. So what kind of indication we are supposed to have? If taking an example of a drug like aspirin is first introduced as an NSAID, right? It is first introduced as an NSAID and now it has been also used as a blood thinner agent like ecosprin at low dose. No, so that kind of indication. So if you are supposed to propose a drug for other new indication, you should definitely have its own roots from its experiential domain. Once you carry that kind of indication from the documented evidence, and you will further establish the same using in vivo and in vitro studies for the establishment of efficacy and safety profile for its newer applicability, right? Then it can go for human pharmacology and phase 2 studies for those findings. So here, what is the other benefit of reverse pharmacology is you can limit the study from phase 2. You cannot right away you cannot go for phase 1 trials also. You can directly go for phase 2 studies because phase 1 studies are almost drawn from the experiential domain, right? And the third major phase of reverse pharmacology includes experimental domains, which involves well-planned experimental and clinical investigations at different levels of biological organizations, extended clinical trials, and subacute safety studies in 2 animal species, right? If at all it is having a kind of other than intended application, through what kind of mechanism it is eliciting the other therapeutic benefit is what established in this experimental phase. Hope I am clear. Right? Coming to the next concept of reverse pharmacology of drugs with new indications. Bedside observations on the established drugs may also have novel response to consider. So this is what is major concern of introducing reverse pharmacology. So once bedside observations are well documented, that could give us a platform to further analyze and further work on the newer intended applications of the established or with drug with much safer profiles. The path of reverse pharmacology can be applied effectively to the studies on new indications for old drugs. So this statement is truly justified in drug repurposing. Hope we are all familiar with this drug repurposing, right? So this is the first and important significant application of reverse pharmacology. So already the drug is available. Already the drug profile is published for a single use. Now, why can't we try the same drug which is already published for its safety, efficacy, and all LD50s, ED50s, everything data or acute toxicity profile, every profile of the drug is already published. So now, what is the novelty in reverse pharmacology is now how can I use all this data to use the safe drug? This is established as safe. No. So now, how can I use all this data and this particular drug to improve the other element? That is what is we call as drug repurposing. Some other terms can also be used as drug repositioning. Right. So drug repurposing or drug repositioning is the basic concept evolved from reverse pharmacology. Right. So the constant vigilance at the bedside may offer much more such opportunities for the enlargement of indications of known drugs. So what should be the ground reality is we should be able to understand every symptom, every parameter to be assessed when a drug is administered. So if everything is completely analyzed, then that gives 100% or holistic profile of that drug. Here, there are certain limitations when you consider in Ayurvedic treatment regimens, unlike allopathic treatment regimens, they also consider patients' diurnal circadian rhythms. Of course, we are now in recent times we are focusing on chronopharmacology, but majority of allopathic medications have least consideration for chronopharmacological aspects, with an exception of certain classes of drugs, right? So, if we are able to document and record every aspect of an individual, see, subject to subject variations are also supposed to be recorded when you take a profile of drug for consideration under reverse pharmacology. So this gives ethnopharmacological, this covers ethnopharmacological aspects of a particular drug which is now re-intended for other applications. Hope I am clear. Here I mean to explain, or here I mean to put forth what I mean to put forth here is a patient's profile. Of course, individual profile about even their height, weight, all the basic parameters we take, all the vitals we take. Other than that, the climatic conditions, the food conditions, the dietary intake, what kind of food the person takes, if it is alkaline, acidic, or junk, even all this matters when you consider the bedside history of the patient to establish that particular drug if it is eligible for taking into consideration to undergo reverse pharmacology for its establishment into a newer avenue. So all this has to be clearly documented. The stigma attached to use of the known drugs for unauthorized indications can be overcome in a fast track by the application of reverse pharmacology to the problem. So this also is a limitation or hurdle for reverse pharmacology. It is not as easy as we discuss, or it is not as applicable as we think to do. It is completely limited and restricted as of now to only traditional medicine or in Ayurveda. So for this to be implicated to allopathic medication, we should first come out of our myth or come out of our boundaries or limitations that allopathic medications, if at all they are intended for other application, it is a kind of unauthorized applicability since it is not established. So in other way, if taking the example of any other drug like we have many allopathic drugs now that are repurposed, right? We have seen in the case of COVID. So taking an example of any drug, for example, an antiviral drug which was intended for application into as only antiviral can also be used as anti-malarial agent. So this we get confined to ourselves that once it is established for an antiviral activity, it shows antiviral activity. If the same drug is given for a person without any viral load in the body, out of our concern or out of our confinement of knowledge, we fix that it should not be given to a person without any viral load in the body. That kind of confinement is because of its is because of our own myth development. See, this is what a stigma developed in our own pharma society, that unintended or unauthorized use of a drug may lead to further damage of a patient. So if the safety profile of a drug is established, then that can be the evidence of its application into a newer therapeutic use or a newer therapeutic applicability. So there are growing incidences of old molecules with newer applications through better understanding of traditional knowledge and clinical observations. So, for instance, botulinum toxin for spasmodic torticollis and hydroxyurea for the induction of fetal hemoglobin. Then aspirin for platelet aggregation inhibition and vitamin K7 for muscle cramps. All these are developed from reverse pharmacology, which we are least aware of. Right? Reverse pharmacology is an age-old process or age-old phenomenon, but still it is not encouraged because it is only limited for its traditional and Ayurvedic applications. And I wish to bring to your notice that there are many allopathic drugs as well. For example, sildenafil. Okay, sildenafil is majorly indicated as an antihypertensive, first developed, and now it has its applicability for erectile dysfunction. So this is also because it has established safety and then it is now repurposed its application. So this is basically evolved from reverse pharmacology. And just like that, minoxidil. Minoxidil is also an antihypertensive drug, first developed as an antihypertensive agent. And now, because it is being toxic when given as oral formulation, it is now because it has elicited an unwanted or adverse effect of hirsutism that was an advantage, and that got developed into a topical formulation for alopecia. So all these have their roots in reverse pharmacology, and now that could be extensively pursued from all our academicians or pharmacologists. So coming to the applications of reverse pharmacology, so major or prime application is now drug repurposing. Drug repurposing is because it could have done much better. It would be much better to start with already established profile of a drug into a newer application rather than developing right from its extraction or right from its chemical synthesis, which would take a longer time than using the knowledge of reverse pharmacology. So reverse pharmacology can help identifying newer therapeutic benefit for existing drug, reducing their development time and cost. Development of new therapeutic agents. Of course, if at all the traditional medicine or folklore medicine insists any kind of extract or a plant or a seed or whatever of a mineral of a natural origin is supposed to have a kind of therapeutic or pharmacological benefit, that also can be taken consideration into reverse pharmacology because it is safer for use, right? The safety of its use may not be documented as a research or as a scientific establishment or as a scientific document, but its prolonged or its a significant use in traditional medicine or a kitchen use. Even a medicine that is intended for its use has a value in it. We have traditional or conventional spices that got isolated into a newer molecule for its therapeutic benefits. So in similar cases, a new molecule can also be developed from a folklore medicine or from a plant which is of traditional use, which is already existed in its traditional use and traditional applicability. So that also reduces the time of development into a formulation, also reduces the cost of development into a formulation. And the new other application is regenerative medicine. And to our surprise, there are many cancer drugs which were developed through the reverse pharmacology. And I'll definitely share the link from which all the presentation was collected and presented with an evidence of publications from PubMed. See, all this research to our pride is done by Indian scientists from CSIR and CDRI Lucknow and as an institute in Mumbai. That only the three organizations together are working on reverse pharmacology, and they are working to that extent that they want every academic institution also to focus on reverse pharmacology, so that this will help in releasing or launching drugs with other benefits which would be so helpful for the current scenario. So reverse pharmacology has its application in stem cell therapy and tissue engineering for the development of newer drugs. So in the sense, this reverse pharmacology has its application in stem cell therapy as well. How could we define it? There is lot much research being done in stem cell therapy for normal application itself. So how could we think of reverse pharmacology has its applicability in stem cell therapy? It is by again using the concept of drug repurposing. So stem cell therapy is basically what we understand is we have a bank of stem cells that get isolated, and these stem cells are banked for its application into either treatment for cancers or treatment for neonatal disorders. And this is what is our conventional approach for stem cell therapy. Unlike our conventional thought process, reverse pharmacology establishes stem cell therapy and its applicability for wide range of infectious diseases as well. So this is yet to be proved, and this is yet to be used. So once it is proved, that could be a tremendous benefit for all the society, because many of the antibiotics have developed resistance, and now we have got an azithromycin-like invention, and through reverse pharmacology, it's a responsibility to launch a kind of drugs that could target major concerns of the society, which are leading to highest morbidity and mortality. Right? So the hurdles what we face in approaching or applying reverse pharmacology are the most of the work is confined to traditional practitioners or academic laboratories and is not taken by industries that are strong in research and development. So here I would like to quote an example of MIT, Massachusetts Institute of Technology, Massachusetts. So it is a 50-story building, it seems. I have not been there, but I have learned it that first 5 floors of that MIT are industries, and rest of the all floors are academic universities and academic institutions that pursue research. So if at all in developing a molecule or in formulating a molecule, these companies or MNCs face any kind of difficulty or any kind of bottleneck that have that were being faced, that were immediately asked by that kind of hurdles were approached to the academic institutions, wherein the solution is brought to the industry is by the students or academic researchers. They offer the solution for the process of hurdle or process of limitation that were addressed during the development. So here in our country or in our scenario of research is not the segment of academia, is a myth that we are living with, right? So though whatever the research that is being done at the level of academic institutions is not further proceeded for higher ends, like higher companies or research institutions to develop into a product. That is the limitation we are facing in our country. So with the same kind of limitation, this reverse pharmacology application is also a kind of limitation, like most of the work is confined to the practitioners, because we don't have enough evidence of documentation for every drug that is being used or every natural product that is being used, right? So we have application of around 70 to 90% of every category of drug has its origin from natural source. In that case, every natural product which has its applicability in traditional medicine should have a vast documentation so far, but we didn't have it. Okay, and because of not able to have this kind of documentation, we could not further explore the other kind of approaches for a targeted drug, right? So lack of documentation, that's what we have discussed, and cultural prejudice. So cultural prejudice is like, so if we by default, we think a natural product, though it is taken, is harmless. But an allopathic medicine, if taken unnecessarily, that causes a kind of damage. So with this kind of cultural prejudice, we are not progressively, or we are not with an intention to explore both traditional and allopathic medicines, we are not approaching reverse pharmacology as first line of approach for drug development, right? So less support from traditional pharmaceutical companies. Of course, this reverse pharmacology still needs to go deeper into the society, or go deeper into the research society. With a correction, I tell that once it reaches all the academic institutions or research institutions, small R&Ds, any kind of institute that pursues research, that would definitely come out with an outcome wherein the applicability will be wider, and in much shorter duration, we could produce many drugs for many ailments which are now much needed. So coming to the further scope and conclusions of the presentation, the current paradigm of drug discovery is shaken by the failure rates of drug candidates at different stages of development. So this is what is major concern now, because investing lots and lots of money, investing lots and lots of time, spending around 15 to 20 years of time on a single drug molecule, if it leads to a failure, that is disaster to call. Like, so target rich and lead poor scenario, we can call, right? Target rich, lead poor. So we are targeting with a rich background, with a much cost input, and we are the outcome is poor, because there is no effective lead molecule with that lead molecule out of this much time invested and this cost invested. So in spite of it, so instead of that, okay, if it has been a reverse pharmacological approach, that would have saved time, or that could have saved money, and that would be at least an eye wiping output. There would be an eye wiping output at least, though not to 100% extent, because it is of traditional background. It could at least elicit any kind of beneficial effect once if it is started. Unless it is approached, it is not addressed, right? The long-standing traditional wisdom of Ayurveda can be explored for drug discovery and development. So this kind of exploration to Ayurvedic medicine or traditional medicine is now of more significance. Even we being allopathic pharmacists also now have to turn ourselves towards traditional medicine to develop the wanted molecules to meet the need. The experiential data can be explored further by relevant in vivo and in vitro experiments. The transdisciplinary approach of reverse pharmacology has sizable scope for new natural drugs and their derivatives. So once if at all we imagine, just to imagine, imagine every ailment has its treatment from a traditional or natural medicine, how cheaper it would be. Because the chemical synthesis of any molecule is costly when compared to traditional isolation and traditional extraction. So in that case, how better it would be if we all focus on investing our time, investing our money into a traditional method, or traditional medicine oriented approach that could be beneficial in all ways, right? Once we establish a kind of molecule out of traditional medicine that could be synthesized chemically using all kinds of course, we have now developed docking studies and metabolomics, proteomics, all this limit time. Unlike conventional drug development, now using all this technology and with enough knowledge of traditional medicine, we could definitely develop molecules which are of high efficacy and safety and can have multiple applicabilities, not just one. So these are the references from which I have presented my topic today. And I would like to acknowledge VV Institute of Pharmaceutical Sciences, Dr. A. Lakshman Rao sir, the principal, and the Department of Pharmacology, Manasa madam, Amina madam, Meeravali sir, and other crew for inviting me for today's session as a resource person. I would like to acknowledge one more thing here is me choosing this concept in presenting in FDPS, in a faculty development program. Why should I select reverse pharmacology as a topic of discussion is because every pharmacology department, if at all, focuses and targets at least one area of research. Like we do it, right? Either a wound healing activity, hepatoprotective activity, whatever. If we select one potential traditional medicine or a traditional isolated molecule and we repurpose or redirect it into another application, that would cause a major significant difference, and of course that would be enough contribution from our side, right? So with this major intention, I have chose the concept of introducing reverse pharmacology into this FDP program. And I hope the introduction of this reverse pharmacology into this session will be helpful both for me as well as to the participants who are present here. So also I thank Hindu College of Pharmacy, my mother institute, for having given an opportunity for me to participate and interact with such a legendary personalities here. Thank you all the participants and faculty.
Thank you, ma'am. Thank you so much, ma'am. Thank you for your sharing insights on the topic, ma'am. We have learned a lot from your presentation. Thank you
Thank
for
you.
sharing your
I
expertise.
thank you. I welcome a kind of discussion if anybody is interested to just discuss of reverse pharmacology, any concepts or anything what we have discussed. If it is really useful for you or if it is really a new input for you, how would you approach this reverse pharmacology within you or within your institute? Any kind of, I would like to listen to any of you people about this reverse pharmacology session now.
Now the floor is open for the discussions. All, if any participant having all queries, can you please ask? Ma'am, I think all the participants is having good knowledge on reverse pharmacology. As well, they are understanding your topic, ma'am. I think.
Oh, that's fine if it is so. If not, also I just wanted it to introduce the same concept because really this is a very novel concept and we could definitely use the same in even designing a very small project for a BPharm graduate or in a PG graduate. So this will help really even for the faculty and the institute to get good enough patents. And I have seen myself and from CSIR ISCT Hyderabad have filed a patent using the same reverse pharmacology knowledge. So this kind of applications will be definitely fruitful for you to also get patent granted. So that's why it's my major concept. So I just wanted to make my presentation so crisp that because reverse pharmacology, once explained within 4 to 5 slides, is completely understood for all the faculty. So, of course, to elaborate with much examples, it was so novel that many of the drugs are not even established completely. As I mentioned, picroside 1 and picroside 2, even that was still under formulation, but it has shown its activity. So I can't just drag my presentation or I just can't expand its avenues for longer time because that couldn't justify. So I need not explain much about the concept because it is self-explanatory if I have explained the basic concept. So that why I just confined myself to give this kind of presentation for all the faculty because that should not be so enough bored. Yeah, is it done, madam?
Ma'am. Yes, ma'am. Thank you, ma'am. Thank you for your valuable time.
Thank you so much. I would like to thank Dr. Lakshman Rao sir for his presence and introducing today and all the participants. Thank you for your presence in the session.
Ma'am, 1 minute, ma'am. We will give you
Sure.
a appreciation certificate for your valuable time.
Thank
I'll
you.
show on the share, ma'am. Please wait 2 seconds, ma'am.
Sure.
Ma'am, is it visible, ma'am?
Yes. Thank you. Thank you so much.
Thank you, ma'am. Thank you so much, ma'am.
Thank you.
Attendance link will be provided on the chat itself. All the participants can be given by today evening only. Thank you.